Okay. Um, so good morning, colleagues. It's very nice to see you here. Let's begin our lecture. It's called Practical Aspects of Radiochemical Purity Determination in Metal-Based Radiopharmaceuticals. Are you familiar with radiopharmaceuticals, with quality control of radiopharmaceuticals? Is it typical for you? So, okay, perfect. You know, this topic is very huge. For me, it's very important and interesting, but in fact, it's impossible to cover all the metal-based radiopharmaceuticals. So I will stop on few examples with few radiometals, which, in my opinion, of course, it's very illustrative. It's very interesting, and maybe it will help you to perform your own research and work, make it better, and just put it on the high level. So in the first part, let's talk about some examples of technetium, gallium, and maybe about the zirconium 89. But first, let's make a focus on the legislation in the radiopharmaceuticals quality control. Unfortunately, in the contrast to the typical radiochemistry with some uranium processes, plutonium processes, and so on, in radiopharmacy, in the field of radiopharmaceuticals, we are under the pressure not only on the department about the radiation safety, but also with many other organizations and departments from the Ministry of Health, Ministry of Production, Ministry of Sales, and other satellite departments. So in radiopharmacy, we have this huge circle of the regulating organs about our production, about our work, about our protocols, and so on and so on. So sometimes when you read some papers and you see, oh, we obtained some very nice molecule, we label it with the radiometal, we inject it in the mice, and we obtain some accumulation in the tumor, for example, we find the good radiopharmaceuticals. No, unfortunately, the situation is not so easy. To inject it into the human, we have to perform a lot of quality tests, a lot of procedure, a lot of the registration stages, just before the first in-human injection. If it would be so easy, I would be very happy, but unfortunately, it's not. Yeah, this slide is about this. So we have the rules and regulations from the radioactive materials, and also we have some big part of rules and regulation processes from the pharmacy, from big pharma, from the radiopharmaceutical production, and so on. The quality is the main characteristic, the quality assurance, because we have a deal with the pharmaceuticals. The quality assurance is a very important part. So we have to be sure that our solution is suitable for injection into the patient in the terms of radiochemical purity, radionuclidic purity, its specific activity, its osmolarity, pH, sterility, absence of endotoxins, and so on. So all these tests should be typically performed before the first injection into the patient. The situation is not the same like with the mice or with some monkey. The quality control procedures or the operation of the quality control typically is summarized in this, let's call it document, summary of the product characteristics. So typically in this brochure, we have all the necessary tests which should be performed before the product release, before the radiopharmaceutical release. And how many registered radiopharmaceuticals do you know? Could you answer me? So far. Radiopharmaceuticals with marketing authorization. Do you know what's a radiopharmaceutical as marketing authorization? Yes. FDG.
Is like a market name or just
No, it's also could be that it's fluoral glucose or something like that. It could be, but it also with the FDA approval is so it has the marketing authorization. Lutathera, Pluvicto, Tauvit, but there's not so many positions in this field. You didn't hear me? Oh, sorry. So there is not so many positions which has the marketing authorization. So the biggest part of the radiopharmaceuticals which used in clinic are produced in the small scale production rules. So we produce it, we make the quality control, and then we use it in our own medical facility. So there is no marketing. In fact, we don't need to have the marketing authorization. And here, there are some interesting reasons. So typically, for the radiopharmaceutical productions which we use in our own facilities, we do not perform all the possible tests. So there is some tricks that we could use just to use these radiopharmaceuticals. Of course, the pharmacopoeia in this question is our Holy Bible, where typically we have all the main monographies which prescribe to us how to perform different tests for the quality control of normal pharmaceuticals and also about the radiopharmaceuticals. So let's talk about the small scale and non-commercial production of radiopharmaceuticals, because this is the biggest part of radiopharmaceuticals which used in the hospitals all around the world. What do we have to do in the quality control of radiopharmaceuticals? First, this identification. Even if you know in your head, in your mind that, oh, in this reaction on the cyclotron or in the nuclear reactor, we can produce this radionuclide, you should be sure that this is the same radionuclide that we would like to use. So this test is very easy. Yeah, we could use the gamma spectroscopy. We could measure the half-life of the radionuclide, and then this is a test for the identification. It's very easy. It's very fast, and there is no big deal with this test. The next test is radionuclidic purity. Also, there's not so many problems just to perform this test. Also, the gamma spectroscopy, typically the gamma spectroscopy. Sometimes we could use the beta spectroscopy or something like this, if it's possible to separate the beta peaks from the radionuclides. But typically, it's gamma spectroscopy, just to be sure that our solution of the starting material of the radionuclide has appropriate radionuclidic purity. pH and isotonic is also very typical test we could perform with some pH strips, also ion exchange. It also is very typical test. So let's not stop on this part. Chemical purity, of course, like all other API, active pharmaceutical ingredients, we have to be sure that this is the same material that we use for the synthesis. And the main part that I would like to talk with you is the radiochemical purity. So in the terms of the pharmacopoeia, the radiochemical purity is the fraction of total radioactivity in the desired chemical form in the radiopharmaceuticals. So we mix our radionuclide with some starting material, with some vector molecule, or with some small molecule, like some chelator, for example. And then we have to be sure that the biggest part, the biggest fraction of our radionuclide, we have it in the desired chemical form. Why it's important? Yeah, it could be the reason of false positive or false negative results of the examination. It will accumulate in not desired in the healthy tissues, for example, in the bones and the bone marrow, blood, liver, or something like that. In the case of diagnostic, maybe it's not very huge problem. Of course, we have false positive, false negative results of the examination, but in the case of radionuclide therapy, for example, if our beta emitter or alpha emitter will accumulate outside the tumor and will irradiate some health tissue, there is a high possibility that this health tissue became the malignant tissue, or even other dose response side effects. So the radiochemical purity is the main characteristic of every radiopharmaceutical. Here, in fact, like radiochemists, like chemists, like analytical chemists, we may use all possible analytical methods just to determine carefully our radionuclidic purity. Most typical method is TLC and HPLC, of course, because it's very common methods. I don't know. I believe in every labs with radiopharmaceutical production or radiopharmaceutical development. You also have the TLC scanner, HPLC machine, solid phase extraction. It becomes common, but it's not very accurate procedure. Electrophoresis, it's some kind of exotic method. For example, I don't know a lot of labs which perform electrophoresis tests, but anyway, you could use all possible tests for your quality control. And if we will look in general notices of the pharmacopoeia, we could see this very nice sentence. For example, in the main monography for your radiopharmaceuticals, it's described that you have to use some kind of thin layer chromatography with a very, very huge development time. Do we really need to perform this test? No, this sentence allows us to perform our own tests if it's provide us the same results or even better results than it's described in pharmacopoeia. It's very nice. It's very important because sometimes, for example, you may not have the appropriate machines or reagents or something like that, but you could validate your own method of the quality control and then perform it in your lab. But the validation is very important. For example, technetium pertechnetate. This is the starting material, the starting radioactive material for all the synthesis of technetium radiopharmaceuticals. And also it could be used like a radiopharmaceutical itself. Do you know where it will accumulate in the body if we inject the technetium pertechnetate into the human? Sorry? In the thyroid gland, and yeah, in the thyroid gland is the last step of the technetium pertechnetate accumulation because it's very similar and it accumulates through the natrium iodide importer, but also you will see the accumulation in the stomach. This is the two main regions of the accumulation, and yeah, it could be used for the diagnosis of the thyroid gland. So if you will see in the pharmacopoeia, if you will take a look in the pharmacopoeia about the radiochemical purity identification of this solution, you will see this test. You see? 2 hours of development. And not only 2 hours of development, just take a look, distribution, radioactivity, dilute, descending paper chromatography. Do you know what is descending paper chromatography? Do you ever see this method in your lab? I believe, I don't know, I think I work in this area for 15 years already, and I never used this method. Because descending chromatography, it's very difficult today just even to find the suitable camera to perform the descending chromatography. Yeah? Because normally we use another way to make a paper chromatography. It's very suitable, it's very easy, it's very fast. What about the descending? But Pharmacopoeia told us about this. Make the descending chromatography for 2 hours just to determine the radiochemical purity of the pertechnetate, not some vector molecule, not some technetium Heinig or Heinig-Tate or something like that. Just the very simple material. Do we really need to use this method? No. Thanks to these general notices, we could change this method to another appropriate, much more suitable method, which is very fast, it's very easy to perform. So with the insending chromatography, for example, with ITLCSG papers. Do you know this one? It's very common in the radiopharmacy. Yeah, it's a glass microfiber impregnated with silica gel. So the chromatography performed in the volume of the plate. It's very fast. I believe for the TLC plates, about 10 centimeters, it takes not longer than 1 minute. 1 minute, 2 hours. I choose this one. I don't know what you will choose, but I will choose this one. And of course, this method is very suitable for the quality control for the biggest fraction of the technetium radiopharmaceuticals. Yeah, and then after the development, after the drying of the TLC plate, we just have to measure the radioactivity distribution on the plate. And here also we could perform different methods. For example, we could use the TLC radio scanner, like Wright test or something like that, like Minigita, for example. Or if we do not have this device, we could measure it with some other appropriative devices like dose calibrator, like gamma counter, like even like with gamma spectrometer. We just have to cut these plates, measure activity of each part, and then they calculate the fraction of the desired technetium form. Typically, the activity of technetium from the generator is about 80, 90 gigabecquerels. So even if you make a very small spot of the solution on your TLC plate, about 5 microliters or even lower, with this activity concentration, you could measure these parts even with the dose calibrator. So activity concentration is very big and it's enough. I believe I saw it when I was in Switzerland. Yeah, they do not perform the test with radio TLC scanners. They just cut the plates, measure it with a dose calibrator, and so on. Then they calculate the radiochemical purity. So this is a picture of how it could be measured, or it also could be measured even with a planar scintigraphy, just with a cyclone or phosphorus imaging. So any possible or suitable devices just to measure the radioactivity distribution could be used in your lab just to perform this test. And also, if we would take a look at all these methods, yeah, typically we perform 2 tests, because sometimes it's very difficult. Technetium, it's very small, it's very active radionuclide. So, and the biggest part of the radiopharmaceuticals with technetium, it's small complexes. So sometimes it's very difficult to separate each chemical form. And typically, we determine the impurity of technetium pertechnetate, which is not reduced, and also the hydrolyzed technetium. Then we determine the sum of these impurities, and then we just, from this 100% just exclude it, and then we obtain the radiochemical purity. But also in your own lab, if you have the good analytical chemist, good radiochemist, you may perform, you may develop your own method. For example, here, for the technetium radiopharmaceuticals, we found very nice system with the separation of all possible forms, with hydrolyzed technetium, with unreduced technetium pertechnetate, and with the main fracture, the main complex of the technetium. Here, the separation factor is not very good, but it's due to the spotted activity. not about the resolution of the system. Sometimes it is not enough to use only 2 chromatographic systems just to perform tests for the unbound technetium and hydrolyzed technetium, but also due to the chemical features of your molecule, you have to add some other system. For example, in the case of Ceritech radiopharmaceuticals, do you know about this? Yeah, it's used for the brain scans with a SPECT CT. Yeah, it's accumulated in the brain. It's very. I really like this radiopharmaceutical because it's metal-based radiopharmaceuticals, but it could penetrate the blood-brain barrier and accumulate in the brain. Here we may have additional radiochemical impurity due to the formation of the secondary complex of technetium and hexametazine, this small molecule, which has no lipophilicity, which will not accumulate in the brain. So we have to determine the amount of this particular impurity, and then we have to add just another one TLC system. It's very, very easy. It's very simple, it's very fast. And then on the sum of these radiochemical impurities, you could determine the real radiochemical purity of your radiopharmaceutical. What about the practical features? You told me that you're familiar with ITLC SG plates. It's very popular. You could open the pharmacopoeia, you could open any article, research article, and then you will see for the TLC quality control in the radiochemical purity determination, we use the TLC plate. There is only one producer in the world which produce this kind of TLC plates. And in our work with gallium radiopharmaceuticals, for example, простите, а как здесь указку включить? Oh, perfect. So we developed this nice system for the gallium radiopharmaceuticals. Here you can see the hydrolyzed gallium, colloidal form of gallium. Here you can see the unbounded gallium. And here, with the solvent front, we could see the labeled molecule. DOTA-TATE, DOTA-TOC, DOTA-PSMA, or DOTA blah blah, any suitable, any popular molecules which are used today with gallium. And this system was validated. We use it, we really like it. But then the situation changed dramatically. And we start to obtain the chromatograms like this. For a long time, we're trying to find the reason of this change in the chromatographic behavior. And then we just take a look at the TLC plates through the light. And you see, there is a different structure of the plates. We just called the manufacturer, ask him about what's happened. And they told us, yeah, of course, we just changed the ref material for these papers, but we didn't receive any problems from other buyers about this. You are the first one who just talk with us about this problem. But yes, this problem, right now we always have the TLC plates like this. And to date, so far, we didn't find other chromatographic conditional just to return in this separation, unfortunately. So in your lab, you could also meet this problem when you develop your own TLC systems, especially TLC system with such type of separation, when we not only have the distribution of the radioactivity at the origin and the front, or solvent front, but also something between these parts. The radiochemical purity determination, it's not only important for the implementation of the radiopharmaceuticals in the hospitals, but also it's very important due to the research and development, of course, because when we're trying to find some suitable conditions for the labeling of our nucleator, of our new molecule, especially with some other new radionuclides, we have to measure the radiochemical conversion. And in the terms of complex formation, we have it's it's almost equal sentences: the radiochemical purity, the radiochemical conversion, the yield of complex formation. Yeah, it is the same terms. So the suitable, the very appropriate method just to measure this parameter, it's important not only in the hospitals, but also in the case of your research and development procedures in your lab and in your department. For example, this is example of new molecule with it's based on the folate vector labeled with gallium. Unfortunately, the starting molecule was very difficult to purify it, just to obtain very pure molecule without some side products. So as you can see here, the radiochemical purity is not very well, even for the mice. And then we just perform some solid phase extraction, for example, with the Oasis Prime HLB cartridges or C18 cartridges or any other type of cartridges which you like. And here we see that radiochemical purity became suitable for in vivo test with animals. And also we see the same picture here with HPLC biodistribution. So of course, yeah, in the hospitals for the patient, radiochemical purity determination is very important, but in your lab, it also should be performed. It will help you to make your research well. Let's talk just a little bit deeper about the gallium-68. I really like this radionuclide because this is the first positron-emitting metal which became in the hospital use, and it promote the renaissance of metal radiopharmaceuticals. It can be obtained from the germanium generator just in the place of your production. You don't need to use the cyclotrons or something like that. So yeah, it's very nice radionuclide. The half-life is just a little bit short, about 68 minutes, but still it's very popular. It could be used for the diagnostics of different kind of diseases. The first radiopharmaceutical with gallium, which we could find in the pharmacopoeia, in the European pharmacopoeia, it's gallium erythritide. It's DOTA-8. This is the same. Yeah. So what pharmacopoeia talk us about this? Oh, of course, yeah. This radiopharmaceutical is used for the diagnosis of neuroendocrine tumors. Yeah? Different kind of radionuclide tumors. So the pharmacopoeia told us that we should use for the radiochemical determination the combination of two methods, TLC method and HPLC method. From the chemistry of gallium, from the radiochemistry of gallium, we know that we may have also three typical form of this radionuclide in the solution. Non-carrier added solution, of course, because typically all radiopharmaceuticals is not non-carrier added solutions of the radionuclides. So the bounded form of gallium, our desired chemical form, and then colloidal form of gallium and free form of gallium, unbounded form of gallium. So we have to determine all these three species at the start. If we do not talk about some specific forms of your molecule, these three species should be determined. And the problem became from the chemistry of gallium because it's very easy to hydrolyze. If the kinetic of your reaction it's not fast enough, if the parameters of your reaction mixture is not suitable, then against incorporation to the vector molecule, gallium will hydrolyze. We will obtain the radioactive colloid. If we inject the radioactive colloid into the patient, we will see the accumulation where? Like all other colloids, technetium colloid, gallium colloid, any other radiocolloid. Come on. Make it clear. Which organ? No. No, it's a colloid. It will accumulate in the liver. Reticular endothelium will filter all the colloids. So if you inject the colloidal form of any kind of radiopharmaceutical, any kind of radionuclide, the accumulation, you will find the accumulation in liver. This is the biggest problem. And in the case of diagnostic radionuclides, maybe it's not a very big problem. In the case of therapeutic radionuclides, yeah, just undesired irradiation of the liver and some other side effects. It's not. And of course, the liver is a very huge organ. If we will see the big enough accumulation in the liver, that will compete with the identification of some lesions that we just really have to find, some maybe tumor lesions or tumor foci. So it could cover the visualization of this foci, because yeah, this is very important, just to avoid the presence of the colloidal form in your radiopharmaceuticals, like all other undesired chemical forms of your radiopharmaceuticals. So in pharmacopoeia, we see that gallium erythritide, the content of gallium erythritide should be not less than 91%. I believe that the biggest part of the world would like to use the radiopharmaceuticals with radiochemical purity higher than 95. 95 is a very typical value for the radiochemical purity. But pharmacopoeia told us 91%. Okay, not a problem. For the gallium impurity B, it's unbounded gallium. We have the limit for 2%. Not more than 2% of unbounded gallium you could find in your gallium radiopharmaceuticals. And for the radiopharmaceutical impurity A, its colloidal form, not more than 3% of the gallium species in then form. And by the way, the colloid will accumulate in the liver. If you inject the unbound gallium, like ionic gallium, where we will see the accumulation of the activity in the body. In the bladder? No. In the bladder, you told me, sorry. In the blood pool, we will see the accumulation in the blood. We will see the visualization of big vessels. And because it's gallium make a very stable complex with the blood transferrin, and that it will start to circulate in the body, and we will see only the blood pool and big vessels. So of course, no specific accumulation also. So if we will take these two limits, and we remember that it's radiochemical purity, it can be higher than 100%. It's a nature, we cannot obtain some additional amount of labeled compound. Then 100% minus 3% minus 2%, we obtain 95%. And here, 91%. Any ideas? That's not coincidence. Where is 4% of radioactivity of gallium? Where we have to find it? What do you think about it? Oh, come on, guys.
Complexes like bronchial molecules, radiolysis species. Gallium 67.
That's gallium 68. Yeah, you're totally right. If you take a look at the HPLC chromatogram of the gallium, yeah, this is, I believe, it's PSMA 617, but just it's the same situation like with DOTA. Here you can see some fraction of unbounded gallium. In HPLC, we never could see the colloidal form because, like in the liver and the body, on the guard column, the colloidal forms will filter. So you will never see the peak of the colloidal form of radionuclide on your HPLC. And here we could see the peak of the desired chemical form. So in this size of the chromatogram, we could see only two forms. But if we look closer just a little bit, yeah, here you could see some additional peaks, additional side products. Помогайте. Тут мышки нет. А что это значит? Ай-яй-яй. Мы не давали на такое разрешение. So here you can see some additional side products, side peaks. It's very difficult to characterize it because there's no standard samples, this radioactive material. What is this amount? This is a peak. This is a peak. This is impurity. Of course, this is impurity. Do we need to characterize it? It's difficult. So this 4% pharmacopoeia gave us for this undetermined impurities that you could find on your chromatogram. That's your radiopharmaceuticals. Тут очень хочет нас записать. За нами все следят. Nope. So here, this is the reason of this 4%, of this uncoincidence in pharmacopoeia. And also let's take a look about the recommendations of some particular methods. If you work with radiopharmaceuticals, you have to be familiar with the system. ITLC plates with ammonium acetate mixture with methanol. It's very old TLC systems. I believe it was created even before the radiopharmacy and radiopharmaceuticals for the separation of the proteins. And typically in this system, according to pharmacopoeia, the labeled molecules have the retention factor about 1. So they're moved with the solvent front. So here we see the fraction of the labeled molecule. Pharmacopoeia also told us that in this system, we could determine the colloidal form of gallium. So here should be the colloidal form of gallium. You see, here, Pharmacopoeia told us for the colloidal form of gallium, you should use the TLC method. For the unbound form of gallium, you should use HPLC method. And then by this combination of these two methods, you could calculate the real radiochemical purity. So we performed these TLC tests according to Pharmacopoeia just to determine the colloidal form of gallium. But in fact, if we will put the unbound gallium on this TLC plate and make a development, we also see that gallium, unbound gallium, not hydrolyzed, also remains in the origin of the chromatogram. So in fact, this is not pure fracture of your one impurity. This is a combination of two impurities together, the colloidal form and unbound form. Pharmacopoeia didn't told us something about this. Pharmacopoeia believes that this is only the colloidal form. And if you will just go strictly to the Pharmacopoeia recommendation, make the calculation, you will obtain the underestimated results of your radiochemical purity determination. So this is another problem, because why do we need the validation of the process, the verification of the analytical procedure in your department? Because you should clearly understand the separation, the distribution of different chemical forms on your plates or on your HPLC system. Because if you will go strictly to the Pharmacopoeia recommendation of any other method recommendation, you could obtain undesired results or underestimated or unclear results of radiochemical purity. And the research and development, maybe it's not a big deal. In the human use of the radiopharmaceuticals, it may provide you very, very big problem. And even now, you could find, a lot of researchers told that gallium and free gallium and colloidal gallium remains in the chromatogram origin, but still, some guys believe that free gallium moves with the solvent front in this system, and very, very nice separation. We can determine the radiochemical purity with this system. I believe that the reason of this misunderstanding is based on the different nature of gallium generators in the world, in my opinion. Maybe there is other reasons. Do you familiar with gallium generator? Did you ever work with it? No? In the world, typically, there are two types of generators of gallium germanium. The first part based on the titanium dioxide, the second part based on tin dioxide. For the titanium dioxide generators, we use 0.1 HCl equivalent. For the tin generators, we use 1.0 HCl as an element. And based on this, based on the nature of the hydrochloric acid concentration is in the eluate, the chromatographic behavior of the gallium could change. For example, in 0.1 molar HCl, free gallium stay at the origin of chromatogram. We just a little bit increase the concentration of the HCl, and then we see that it moves with the solvent front. So maybe here we could find some problems of this misunderstanding in some articles, in some pharmacopoeia monographies, and so on. It's I really like this example. I believe for many years I always talk about it to my students on the lecture. Misunderstanding could be found in the pharmacopoeia. This is USP, United States Pharmacopoeia, and monograph for the technetium hexametazime, Seretec for the brain visualization. And for some radiopharmaceuticals, we have to perform also biodistribution test. We're trying to avoid it, but sometimes we have to do that. Just before the injection to the patient, we have to inject it to the rat or into the mice, measure the distribution of radioactivity between the organs, and then just to prove the real suitability of the radiopharmaceuticals. For the technetium hexametazime and biodistribution tests, Pharmacopoeia told us, make injections, sacrifice the animals, carefully remove the brain, liver, intestines, including gallbladder. But the problem is that rats don't have gallbladder. The nature didn't give them the gallbladder. But Pharmacopoeia told us to do that. So if you read a lot of Pharmacopoeia monographies, you could find a lot of this nice moments, for example. But anyway, the Pharmacopoeia is our holy Bible, so let's follow it in general notices and all other recommendations. So, as you may perform your own test, you may use alternative methods. For example, I could recommend you to use this system. It's very simple. You don't need to use ammonium acetate to purchase ammonium acetate, the suitable purity of the suitable quality. You just have to use methanol and normal saline, make a mixture 5:1, and then you will obtain almost the same separation factor. Here, the retention factor is not 1, it's 0.8, which is much more suitable in terms of chromatography. Because when we have the resolution with the retention factor about 1, the resolution of your system is acceptable. So it's not very good in terms of chromatography. This kind of separation is very nice. And here you see it's very fast, 4 minutes versus 20 minutes. It's very easy to perform, and it's suitable for a big part of gallium-based radiopharmaceuticals in your lab. But also, this system is just a little bit Fox-like. We have also the same problems if you use very high concentration of hydrochloric acid in your starting material, then you can see these changes in the chromatographic behavior of the gallium. So you should keep in mind these problems which you could find. Also, as you may develop your own method, not only follow Pharmacopoeia or some other articles, for example, we develop our own method for gallium-based radiopharmaceuticals, and we obtain the distribution of this. Here you can see the colloidal form of gallium, here you can see the unbound form of gallium, and here you can see the labeled form of gallium, desired chemical form of your radiopharmaceuticals. This is the model composition just to show you all possible impurities, and this one is the real preparation of gallium with the suitable radiochemical purity for even for in vivo use or for the human use. And see, this is the retention factor for all 3 gallium forms. And the system is very easy. Also, ITLCSG plates and just a mixture, just a water solution or 3-fluoroacetic acid. It's very typical reagent in any lab, in any analytical lab, in any radiochemical lab. Yeah, and this system is very suitable for different type of this popular radiopharmaceutical DOTATOC, DOTATATE, Nadagenok, RGD dimers, for the PSMA 617, also for the folate-based radiopharmaceuticals. So if you work with gallium radiopharmaceuticals, I recommend you to try the system in your lab and perform some tests just to be sure that your preparation is good. And also in the processes, why the validation is very important. Just before implementation of this system in our practice, we have to perform a lot of suitability tests. So here you can see the pharmacopoeia method, another pharmacopoeia method, our method, and here you see the full coincidence of all parameters in the determination of impurities, and even with electrophoresis, just to confirm that its system is good, provide us real distribution on the forms, and after that we just start to use it in any other kind of radiopharmaceuticals based with gallium. There are some problems because pharmacopoeia recommend us to use HPLC, liquid chromatography, to determine the unbound forms of gallium, or today for even other radionuclides like lutetium or something like that. If we compare the results of TLC and HPLC methods, what we will see? Here you can see the TLC results. Yes, some amount of colloidal form, some amount of unbound form, 94% total radiochemical purity. With HPLC, you can see 98%. So if we exclude colloidal content, we will take 98%, 98%, good, yeah? But then we take this model preparation with some amount of colloid, a little bit more content of unbound gallium, some amount of the desired chemical form. This radiochemical impurity, because it's impure, it's 78%. Then we inject into the chromatograph, and what do you see? 98%. What's the problem? The HPLC told us that our radiopharmaceuticals, it's suitable for in vivo use or in human use or for other studies. But TLC told us, no, it's not, absolutely not suitable, 78% of desired chemical form. So it was found that non-carrier added forms of metals, of radioactive metals, which we use in our radiopharmaceuticals preparation. Typically, it's 3+ metals, are unspecifically sorbed on the reversed phase in the HPLC column. The mechanism is unclear. This process is unclear, but we have to keep in mind this fact that unbound form of your radionuclide may sorb, and it became the higher the pH of your preparation, the higher the sorption of your radionuclide on the column. So if you did not perform some additional manipulation with the radioactive solution or with your HPLC method, you could see the picture like this. So yeah, this is a typical area of the pH of the intravenously injected radiopharmaceutical. And as you can see here, 90% of the 90% of your unbound fraction will sorb on the column. So you will never see the fraction of unbound radionuclide with HPLC method. So you should keep it in mind and not trust this method only. Never exclude the TLC method from your quality control methods for your research or for the quality control of the radiopharmaceuticals, because it's very popular when you open some articles, research articles, and you see the materials and methods section, and then you find radiochemical purity. We perform this test with HPLC, blah, 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 Shimadzu or something like that. No HPLC test. It's very simple. It's very fast. Put some spots on the TLC plate, perform it, just make a development, just to be sure that there is no colloidal form, that there is not a big amount of unbound gallium or something like that. And then just say that, okay, the radiochemical purity is good of your radiopharmaceutical. This is the difference between methods. The mechanism of this process is unclear, but just if you would like to come closer to this mechanism, we could simulate this biodistribution and, for example, use also TLC plates with reverse fast coated TLC plates. And here you can see that with increasing the pH of your solution, you see the retention factor became lower, and also in here in the natural conditions, the free gallium stays at the origin of chromatogram. So this is very similar to the processes of the liquid chromatography during your HPLC tests. What we could find in the literature, for example, okay, after my lecture or maybe in your own research work, you will find some articles like this, where we can see GMP compillant, gallium radiolabeling, blah, blah, blah. And here you can find this nice picture with a good correlation between TLC and HPLC. Then you remember, oh, in Moscow State University, Anton told us that there is no coincidence between this method. And here we could see this coincidence between this method. What's the problem? If we will go to the materials and methods of this kind of articles, when you can see this nice results of coincidence, how they perform the addition of the unbound gallium impurity. They produce the gallium NOC, then purified it with the C18 cartridge, then reformulate it in the normal saline, and then add unbound gallium from the generator eluate. So we have the saline, we add the even 0.1 molar HCl. What's going on with the pH? It's decreased. And if pH decreasing, of course, I show you the coincidence became more thin, so the sorption is not so high. But in real preparation with the real pH with the stable pH of your solution, you will never see this kind of correlation. Never. The free gallium will sorption on your column. But still, we cannot exclude HPLC tests also, because it's very nice method for the determination of the side products of your own molecule, degradation products of your, or even radiolysis, of course. So we cannot exclude HPLC. Is it possible to rehabilitate the HPLC method in terms of the unbound gallium or unbound radioactive metal determination? Due to some model experiments, the first idea what we have to do, it's just increase the acidity of your eluate and the HPLC. Yeah, and here you can see that distribution, the retention factor of unbound gallium stay the same in all area of pH possible in your radiopharmaceutical preparation. But the problem is that today HPLC columns, the pH range just to perform the analysis with these columns, it's limited. It's quite limited, and we cannot use 1% 3-fluoroacetic acid just to perform this test. It will destroy our column. Or one column for one analysis, I believe it's very expensive, and not everyone ready to do this kind of test in your lab. So no, this way it's not for us. Another one, it's very suitable, for example, is the sample preparation before analysis. You can take a sample of your radiopharmaceutical preparation, add some chelator. For example, DTPA, DOTA, it's not suitable because you have to reheat your mixture, or some exotic chelator like HBD or something like that. It will form the complex with unbound gallium, and then you inject it into your HPLC system, and then you will see the real distribution of the radioactivity forms in your solution. But also, if we perform experiments with some new kind of chelators, not typical DOTA or DAGA, DOTA, BATA, or something like that, you create your own chelator. So you have to perform the labeling tests, stability test, and then if you will add another one chelator, they will start to compete. They may compete in this reaction. So also your results will be false positive or false negative. So it's not suitable for all cases. For typical molecules, yes, you could make this sample preparation methods. For new molecules, it's not very good. So they are, oh, sorry, it's in Russian. The other method is just to add some soft complexon in your HPLC element. DTP, it's not suitable because it's very difficult to dissolve it in acetonitrile. In water, it will dissolve. In acetonitrile, for example, it will not dissolve. And then we just think about just very, very easy complex formator. Complexon, it's like citric acid. Прости, пожалуйста. Прости, а можно вот убрать вот это верхнее, потому что закрывает почти все слайды. Ну, хоть куда-нибудь. А то там название. Пусть будет там. So, the citric acid very easily form complexes with gallium and with a lot of other radiometals. If you work with radiopharmaceuticals, you know the system ITLC citrate buffer or ITLC citric acid buffer when unbound form of radionuclide moves with the solvent front. Also, we may use this reagent with this nice substance in your HPLC system. And then... А теперь не переключает. So with this small modification of your eluent, you may obtain very nice recovery of activity from your HPLC-18 column. And then, because of complex formation between gallium and citric acid, there is not big competition between citric acid and good chelators like DOTA or something like that. And also, there is no sorption of your radionuclides on the column. And then you may find this nice correlation between your TLC methods and your HPLC methods, and you see the declines about 45 degrees. So yeah, it's a total coincidence and validation. It's a good validation parameter of your new method. And there is no big error between the TLC determination of unbound form of radionuclide and HPLC form of your unbound radionuclide. Of course, sometimes we have some problems due to the nature of the molecule. You see, this is the three very popular forms of gallium octreotide analogs which used in diagnosis of neuroendocrine tumors. It's TOC, TATE, NOC, and almost all of them is similar. Here you see the beta-naphthyl alanine through the tyrosine and so on. There's not big differences in this molecule, but chromatographically, you may see this huge difference in biodistribution and retention factor of these molecules. It looks similar, but here you can see DOTA-TOC or DOTA-TATE had the retention factor about 0.5. But in the case of DOTA-NOC, very popular molecule in radiopharmacy and radiochemistry, the retention factor is very low. So here in this particular case, it's impossible to separate the colloidal form and labeled form. So if you make a deal with this kind of molecules, you have also perform some additional TLC test or just choose another system to perform this labeling parameters, just to perform your radiochemical purity determination test. And with other radionuclides, for example, here with lutetium, we also see that here with lutetium, the shape of the peak is not so narrow, it's not very nice for determination. So here, for example, citric acid eluent and also ammonium acetate may be used for the determination of the radiochemical purity of your radiopharmaceuticals. And also, there is another one interesting point. If we compare the chromatographic distribution of gallium with lutetium, this system is completely unsuitable for your analysis because unbound lutetium don't even have the straight retention on the chromatograms, just spreading on the chromatogram, and you will never determine the unbound fraction of your radionuclides. So here, if you have some unbound fraction, it will be spread. It will spread like that, and it's very difficult to quantify it. Oh, there is example. Here, the TFA element. This is a labeled form. This is unbound lutetium. This is unbound lutetium. This is unbound lutetium. We perform the pharmacopoeia test with ammonium acetate, and here, this is even not the peak. And it's very difficult to determine. It's very difficult to measure it. So for lutetium, this system, it's unsuitable. Why I'm talking about it? Because very often you could see in some articles that people perform this test just to be sure on radiochemical purity, but it's unfortunately, it's not suitable. If your composition is okay, then you will see the nice chromatogram. If your composition is not okay, you will also have this nice chromatogram. So never make this mistake in your lab practice. Let's take a look in other new monography of gallium radiopharmaceuticals. For example, gallium gosetitide injection. Do you know this one? This guy? PSMA 11. It's just PSMA 11, the very popular molecule for diagnosis of prostate cancer. This is another monography which we could find in Pharmacopoeia for gallium-68 radiopharmaceuticals. And here already we have some much strict limits for the determination of the gallium, unbound gallium. Sorry, it should be, you see, limit, not more than 3% of total radioactivity is due to the gallium species merging with retention factor more than 0.2. So the sum of unbound gallium, of colloidal gallium, in the case of this particular radiopharmaceuticals, is already not more than 3%. If you remember the situation with DOTA, we have 3% for colloidal form, 2% for unbound form, 5%. Here already 3%. Why? I have no idea. Maybe because this big gap between the time of releasing of this monographies, because the DOTA monography is, I believe, 7 years old already. This one is just 1 year already. We became more precious, more accurate in our production, so the limits become strict, more and more strict. This is only one reason for me about this. And here we can see another one problem due to the species of your own molecule. If we inject our radiopharmaceutical based on gallium PSMA 11 into the HPLC LC chromatograph, you will see this kind of distribution. This is not one peak, this is 2 peaks. And what is the reason why we see this picture? Because of the chelator used in this structure, used in this pharmaceuticals. It's very nice chelator because it may form complexes with gallium without heating. It's very unsensitive to the metal impurities. So it's very difficult to obtain bad radiopharmaceuticals and with gallium in the case of this chelator. But also we have some problems with diastereomers because it may form 3 forms of diastereomers in your reaction mixture with different thermodynamic stability. And here In this case, we have to determine the amount of each complex. Which diastereomer form is better for the clinical practice? This is an open question because thermodynamic stability is different, but a lot of studies show that biodistribution is almost the same. So yeah, this is a problem for analytical chemists, how to validate, how to perform method with two peaks and how to make the real calculation. The solution may be just in the changing of your synthesis procedure, because if we hit the reaction mixture, then the amount of the more thermodynamically stable complex will increase and this side peak will decrease. So you will see almost one peak on your HPLC chromatogram. If you not hit your reaction mixture, you will see these two forms. So you have to validate the method with two peaks with your analytical signal. Another problem why we cannot exclude HPLC method. Yeah, and of course, these two forms of diastereomers, you may see it only with HPLC. You will never see it with TLC methods. That because we cannot exclude HPLC method. Another problem is radiolysis and other kind of degradation of the vector molecule, because typically, just to make a labeling of radiopharmaceuticals with gallium, we have to perform some post-processing procedure. And for example, it may be acetone-based method, ethanol-based method, and so on. And in the case of acetone method, we perform the labeling in the mixture of acetone, gallium, HCl. And this mixture provides the degradation of the molecule. So we have to perform, we have to determine the amount of this degradation products. And also, even that in the case that gallium-68 is beta plus emitter, we may see the presence of radiolytical degradation. Even in that kind of radionuclides, we can see the products of the radioanalytical degradation. So even in the terms of research and development and also in your regular production, we have to perform the HPLC test just to be sure that amount of this side product, side radiolytic impurities, it's suitable for in-human use of your preparation. And also with TLC, for example, it's very difficult to determine the presence of radioanalytical degradation. Yeah, here the peak of your labeled form became not so nice. Yeah, it's not even the peak. Yeah, but it's not enough analytical signal just to clearly say, yeah, this is the problems of the radiolysis. They may be another problems. Okay, let's stop about this kind of correlations. Let's talk about this very nice radionuclide. Do you know something about zirconium 89, sorry. It's very popular radionuclide. And I also really like it because the half-life of zirconium is about 4 days. This radionuclide is very suitable for the diagnosis with monoclonal antibodies and fragments of monoclonal antibodies, because monoclonal antibodies has very high period of circulation in the blood, so we have to wait enough time just to see the accumulation in the tumor foci. Normally, it's about 24, 48 hours post-injection. And zirconium with the half-life of 4 days is very suitable for this kind of processes. And it's very easy to obtain this radionuclide. We do not have to use some unreached metal targets for the cyclotron. We used yttrium, it's a monoisotopic element. We just irradiate it with protons with not high energy, just extract it, and then we could make the synthesis of our radiopharmaceuticals. In the case of zirconium, very typical method to determine the radiochemical purity is TLC with the solution of DTPA with a pH 7. In theory, DTPA make a complex with your unbound zirconium, just move it to the solvent front. Your antibody, because it's a very huge molecule, always will stay at the origin of your TLC plate, and then you will find this kind of separation. Here is a labeled antibody, here is unbound zirconium. This is example of very good prepared radio-labeled antibody. This is our results. Not very good, because we're trying to perform just to repeat these tests, but we never obtained some such good separation results. But there is another problem based on the nature of the starting solution of your radionuclide. Typically, just to extract zirconium from the irradiated target, we used hydroxamic acid-based radiopharmaceuticals. If we use hydroxalic acid-based radiopharmaceuticals resins, sorry, if we used hydroxamic acid-based resin to extract zirconium, then just to recover our radionuclide, we have to use oxalic acid solution. And if we compare the results of the distribution of zirconium in the media of pure hydrochloric acid and hydrochloric acid, which contains some amount of oxalic acid, we could see that the distribution is completely different. And this difference based on also on the complex formation between zirconium and oxalic acid, and then we have the problems with the competition between oxalic acid in the processes of the incorporation of zirconium into the molecule. But as you can see, all these chromatograms is very difficult to measure, to calculate, to determine which fraction is unbound zirconium, which fraction is labeled amount of zirconium, which fraction is oxalate complex, which fraction is chloride complex, and so on. So we found some other system, for example, which could provide us some distribution of zirconium, which looks like very promising and very and really correlates with the speciation of zirconium in the water solution. And also we just compare it with this typical system with DTPA. As you can see, the difference is huge. But what does it mean? In fact, we could say, oh yes, this is an unbound zirconium. This is two forms of unbound zirconium. This is colloidal form of zirconium, but here you could see the unbound form of zirconium. This is colloidal, and here we could see colloidal and free form of zirconium. What is this? How we could identify these forms? And here, the biodistribution tests may help us just to determine these forms of zirconium. So the first, if we inject this solution, you see these two systems, they both show us that we have the only unbound zirconium in the solution. We see very typical biodistribution of zirconium in the body, because zirconium accumulates in the bones after 20 hours post-injection, but in the final, it will accumulate in the bones, and here is the grow area, and so on. In the case when popular and typical system with DTPA told us that there is only colloidal form of your zirconium, our system shows that it's still unbound zirconium. We inject it into the mice and see this is typical biodistribution for unbound zirconium. There is no colloid like DTPA system told us. If we inject this model solution when we have just a small amount of unbound zirconium, just colloidal zirconium, big amount of unbound zirconium. DTPA system still told us that it's only colloidal zirconium. Here we already see accumulation in the bonds and some small accumulation of colloidal form in the liver. Yeah, it's very nice. I believe there's a very nice correlation between biodistribution and your analytical methods when you see by your own eyes this, that your results really in a good agreement with biodistribution. And finally, if we use, when both systems told us that there is colloid, this is colloid. This is only the liver. There is no biodistribution in other organs in your laboratory animal. So, also in your work with, if you will work with zirconium, for example, I strictly recommend you to avoid use of this system with DTPA because it provides you false positive or false negative results, and it cannot provide the real amount of active forms of zirconium in your solutions. And try to use alternative systems, our own system, or you can create your own system, validate it, and then use in your tests. And before the break, I believe I have a few minutes. In radiopharmaceuticals, we have another one problem. What is the sentence for the radiopharmaceuticals? The fraction of radioactivity in desired chemical form. But what is desired chemical form? In fact, we work with radiometals, non-carrier added radiometals, when it's very difficult to determine the real chemical form, because nuclear resonance, EXAF, Xanax, they cannot provide us suitable data about that. Yeah, for example, zirconium in analytical concentration become polymerized, and nuclear magnetic resonance will show you polymeric forms of zirconium. But in radiopharmaceuticals, a non-carrier added solution, the concentration is so small that there is no polymerization. This is a different kind of chemistry. This is different kind of speciation. And this is just a very nice example. If you're familiar with the bone scintigraphy, it's very popular procedure for patient with cancer, yeah, because very often cancer make metastasis in the bones, providing very big pain. So I believe every cancer patient should be done with this bone scan. And MDPI, methyl diphosphonate, it's a very popular pharmaceutical with technetium just to perform this test. But what is the real chemical form? We told, pharmacopoeia told us the undesired chemical form. What is desired chemical form? Of course, in the terms of, for example, thin layer chromatography, we may perform one peak, okay? And we say, this is our desired chemical form. But chemistry of technetium, in fact, is much more difficult. And if we perform the, for example, electrophoretic separation, we will see that there is 4 forms already of your radionuclide conjugated with your small molecule. But all these forms will accumulate in the bonds. So the question is, what is desired chemical form? All of them. But pharmacopoeia told us form, not forms. You see? So we call it some lacuna and misunderstanding of these terms in radiochemistry, or this concentration of radionuclide. Pharmacopoeia told us form, but we have forms. It's different forms, but they all accumulate in the foci. So sometimes it's your own opinion, your own choice, how to perform this quality control test and which form you should, you will believe that it's your desired chemical form. So let's make a break for 10 minutes or 15 minutes. 15? Okay. component. So let's continue. In the second part of this lecture, I would like to talk with you about two very popular radionuclides today. First is lutetium-177, and we will talk a little about actinium-225. Lutetium, very popular radionuclide. It's used in targeted radiotherapy of different diseases. It has very nice nuclear characteristics to perform this radionuclide therapy. And today, in the world, we have already two radiopharmaceuticals with marketing authorization. It's Lutathera and Pluvicto. So you're already familiar with these two molecules. This is the same dotted date for neuroendocrine tumors and PSMA 617 for the prostate cancer treatment. Yeah, because it's not enough just to visualize the tumor foci and say the patient, okay, you have the disease on the third or the fourth stage. We have to try to help him. Yeah, and the radionuclide therapy is just a very nice way to perform this treatment. So, and of course, yeah, because the nuclear properties, lutetium is very popular, it's very common and very useful radionuclide for radionuclide therapy. And as I told before, in the case of diagnostic radiopharmaceuticals, the problem, if you determine the radiochemical purity incorrect, the only problem is the false positive or false negative results of the examination. In the case of the nuclear radiotherapy, incorrect radiochemical purity may provide side effects due to the undesired irradiation of false tissues, or the tumor foci will not obtain desired dose of the radiation just to obtain the good response to the therapy. That because radiochemical purity and therapeutic radionuclides, it's very, very important. But before, let's take a look on some examples of these radiopharmaceuticals. PSMA INT is just another kind of PSMA ligand for the therapy of prostate cancer. It's very popular. It's out of the patents. So many centers all around the world very like to use this one, this molecule for treatment of the patient. This is the radiochromatogram of lutetium PSMA INT preparation. What do you think about this results of the chromatography? Is it good or not? Yeah, this looks good. It's just a little bit asymmetric, but there is no side peaks. Just you can see only one peak, just a little bit narrow. It's narrow enough. So if you see this results of the chromatography, you may say that, okay, the radiochemical purity of this preparation is good enough, so we could use it for the patient. And this one, not so good already. Yeah, here you can see some side peaks close to the main peak of your molecule. It's still good, but the background, it's not very good. But also you see this side products of the synthesis of your molecule. This one, completely bad already. Yeah, a lot of side products, even with a very narrowest sharp peak of the main chemical form. And here, disaster situation. Yeah, it's absolutely unsuitable for the, if you see this chromatogram, it's unsuitable to inject it into the patient. But the problem is that all these chromatograms for the same preparation. You see, the results of your quality control, it's very sensitive to the method that you choose to determine the radiochemical purity of your preparation. We just compare the different methods published in the literature, monographies, just make this, let's call it standard preparation of the radiopharmaceuticals. And as you can see, the chromatographic picture is very, very different from one method to another one method. And also, we could see this not only with PSMA-INT. If we will take this popular molecule with PSMA-617, also the situation changed from one method to another method. In one method, you could see only one peak. In another method, you can see 4 side peaks, one side peak, and some additional peaks at the beginning of the chromatography. So the situation, the chromatographic picture changed dramatically. So where is the truth? In fact, if we establish the suitability, acceptable limit of the radiochemical purity of your radiopharmaceuticals around 95%, it's a very common value for the radiopharmaceutical, and then compare obtained results with different methods, you can see this discordance from very acceptable results to completely unacceptable results for the same radiopharmaceutical preparation. And this discordance is a disaster. So I can tell you, for example, that if we will return, this chromatography that you told me, it's very nice. It could be injected. I believe it's from the Milano University of Health, the Iorio group. They used this method just to analyze the radiopharmaceuticals, and they inject it into the patient, I believe, every day. But then, if you compare, you can see that in some cases, the real radiochemical purity could be less than acceptable limits established even in your own established limits in your radiopharmacy facilities. So what's the problem? Even if the situation could be observed not only with lutetium radiopharmaceuticals, for example, if we compare some pharmacopoeia HPLC method for gallium radiopharmaceuticals and then compare it with some other good developed method, we can see that here with the pharmacopoeia method, we see only one peak. It's not very narrow, it's not very good sharp of this peak, but still it's only one peak. And we could say, yeah, it's suitable for the injection or for other in vivo use. But here, with some good developed methods, you can see a lot of side products, and you could say that the real HPLC radiochemical purity of your preparation is not suitable for any kind of biodistribution studies or for in-human use. And keep in mind that we make a deal with beta minus emitter, the radiolysis problems increased. And when we're trying to analyze the radiolytic degradation of the products of these radiopharmaceuticals, the discordance between different methods became even higher. Here, the starting material, here, after a few hours of radiolysis, you can see this discordance between the radiochemical purity values in your preparation. That because the choice of the appropriate, suitable method for analysis, it's very important. And because even for lutetium preparation, there is no monography in pharmacopoeia in the world. There is, if your hospital or your department not work with Novartis, you do not buy the Lutathera ready to inject radiopharmaceutical or with Pluvicto, you have to perform the synthesis in your own lab. So you have to perform the quality control in your own lab. Then you have to develop your own test methods just to perform this quality control procedure and also to determine the radiochemical purity. So the determination of radiochemical purity in that case, it's on your own responsibility. So the choice on the method is your choice, like chemist, regular radiochemist, like analytical chemist. And because we make a deal with the patient, it's very important. What's the reason of this disconcordance? For example, in the case of PSMA ligands, very popular all around the world. I believe today the prostate cancer is on the second, or yeah, I believe it's on the second place in the reasons of men's death all around the world. So yeah, this radiopharmaceuticals is very important and very popular. So what's the reason of this disconcordance between methods? First is the side products of the synthesis, because when we add the radionuclide to our precursor, just to make the incorporation of radionuclide as full as it possible, we have to heat. Yeah, normally we have to heat the reaction mixture around 80 or 90 degrees Celsius. And it was found that during the heating of your reaction mixture, we can observe the formation of these three side products. This side products is based on the spontaneous cyclization of the vector fragment of your molecule. So you just put some heat to your reaction mixture, and then your vector molecule became to change. And all this forms, all these side products of the heating of your reaction mixture is unspecific to the PSMA receptor. So this forms, it looks very similar to the starting molecule, but they will not accumulate in the tumor foci. They will just wash out from the body or accumulate in some undesired parts of the body. So yes, of course, we have to limit the amount of this forms during synthesis and during your analysis of the radiopharmaceutical. So here was a very nice article which showed that all these three forms is unspecific to the LN cup cell culture or even to the prostate cancer foci in the body. So they just wash out. So we didn't reach the desired amount of activity in the tumor foci. So there will be no response to the therapy. In our work, we also found another one, impurity. Here you can see that it's the amount of this impurity, it's not very high, but this impurity, this is a complete destruction of the vector fragment of the molecule. Here we have only the chelator, the linker, and just the linked part. And also this impurity may form during the heating of your reaction. And sometimes, depending on your method, all these three forms may just became to the coalescence between the main peaks. So typically you could find this only one chromatographic peak, which in fact contain from 1 to 5 already different chemical form of your radionuclide. And this is another example because our lab, the quality control lab, we also perform analysis to another facilities, another department, and one producer provide us the sample of PSMA just to perform the quality control test, told us, oh, with the TLC, we have absolutely perfect radiochemical purity. We have plans to inject it. We performed the HPLC test, and as you can see, no, it's not suitable for human use, of course. It depends on the synthesis problems, it depends on the analysis problems, but maybe this situation is better than nothing. Just to, I don't know, not to perform any help to the patient. But still, if we talk about the nuclear medicine, high precision medicine, this kind of radiochemical purity is not very good for the use. Another way, another reason of this disconcordance is your own apparatus that you use in your lab, because depending on the volume of the measuring cell in your HPLC, you can see the changing in your chromatograph. The volume may be very small, and then you have this very nice separation with all three possible forms of your preparation. Then, if you use a little bit higher volume of your analytical cell in your HPLC detector, then you also will see the coalescence of the signals, and then it will be one chromatographic peak, which could be analyzed like only one chemical form, and then it will be underestimated results of the radiochemical purity determination. Another one reason is the software parameters of your HPLC, because sometimes the analytics even not put some specific parameters to the measuring of the peaks. And for example, if the parameters of the software put on the 5-point adjustment for each signal, you will see the very nice resolution for the peaks. If you put on the adjustment to calculate the average value of the signal for 25 points, you will see also one signal. Yeah, it's kind of smoothing of your signal, but you will never separate these peaks with this kind of smoothing. So this parameter is very important when you perform the analysis of your radiopharmaceuticals also. Not only the method, not only the column, not only the element, but also parameters of your software and parameters of your measuring cells in your radioactivity detector. And also, yeah, we have the problems. Another problem is the radiolysis. Yeah, this very nice example. This is a model preparation of PSMA with radioactivity concentration close to the clinical. It's about 5.1 gigabecquerels. And even if we do not add some stabilizers of radioprotectors to the reaction mixture, already after 30 minutes, typical time for the synthesis, we see this very bad picture of the chromatography. We see a lot of radiochemical impurities, and in fact, only 54% of the radioactivity is associated with your main desired chemical form. So the radiolysis is another problem of the determination of radiochemical purity of therapeutic radiopharmaceuticals. So here is just a little bit closer, this chromatography. Accepting these 3 peaks of thermocyclization, you see 1, 2, 3, 4, 5, 6, almost 10 side products due to the radiolysis of your molecules. So you should analyze it, you should perform the analysis just to identify them, qualify them, and then maybe put some optimization steps in your synthesis procedure. That because analysis is very important also at the stage of development. What I would like to talk about here. Oh, yeah. And another interesting option in the lutetium analysis is, as well as in the case with gallium-68, you should be very careful when you're trying to perform some published methods in your own lab. Here is a very nice example. This method was published, I believe, 1 year ago, and it based on the phosphide buffer just to improve the recovery of labeled form from the chromatography. But the recovery of labeled form from a chromatograph was very well. There was no retention of the labeled form in chromatograph. But if you add some amount of the free lutetium, unbound lutetium, in this method, do you see unbound lutetium? No, but this preparation contains it. So you should be very careful when you're trying to perform published literature data in your lab. If we read this article very, very carefully, there is just some small note that all preparations contain DTPA also. In the case of DTPA, yeah, we will see the same situation with unbound lutetium like this. We will see the peak of the unbound lutetium, but depending of your standard procedure, of your synthesis procedure in your lab, maybe you don't want to add some DTPA in your radiopharmaceutical preparation, because if you add DTPA, then you have to quantify the DTPA in your final preparation. Yeah, it's additional analytical control. Do we have to add it or not? It depends on you, but I'm just talking about that. In the case when you do not add some additives in your radiopharmaceutical preparation and trying to perform some published test, you may meet this kind of problems. Because, yeah, these preparations contains this huge amount of unbound lutetium, but here with this phosphate buffer method, we cannot see them. So we could say, oh, the preparation is very good, we could inject it. In fact, no. And also, I already talked about it in the case of the TLC analysis of lutetium radiopharmaceuticals. If your preparation with this very popular method, just be careful when you're trying to use this method in your practice in your lab. If the preparation is good, you will see the one peak, which corresponds to the labeled form of your molecule. So we could say, yeah, the radiopharmaceutical preparation is okay. But unfortunately, all the radionuclides from the rare earth elements group, except scandium, it's yttrium, terbium, thulium, samarium, lutetium, in this system have absolutely different chromatographic distribution, and unbound form migrates with the solvent front. So as I showed you before, if you have some amount of unbound radionuclide from this group, especially this therapeutic radionuclides like yttrium, like lutetium, you will never determine them with the method of the ammonium acetate and methanol mixture with your TLC system. So it is always better to perform your own methods in your lab. From the practical reasons, I may recommend you few systems. For example, of course, very typical method with we talk about it already with citric acid. Here, unbound form of lutetium will migrate with the solvent front, labeled form will remains on the origin. In the case of lutetium and related radionuclides like terbium, like thulium, we do not have the problems with the hydrolysis. If gallium very easily hydrolyzed and form colloidal form, it's very difficult to obtain a radioactive colloid of lutetium. So just don't think about the colloidal form of your radionuclide. Of course, if you perform your synthesis not very carefully, we may observe pseudocolloidal form of radionuclide in your preparation because of dust or because of some small particles, but it's very easily, it may be easily removed just with a filtration. So the chemical reasons of the colloid formation for lutetium, it's very difficult to perform it. So in that kind of radiopharmaceuticals, we'll only talk about unbound lutetium and conjugated lutetium. So this is two systems very suitable, in my opinion, for analysis of lutetium preparation. And also take a look at these two systems. If one day, is anybody works with lutetium already? No? No. One day, if one day you will start your work with lutetium and analysis of lutetium preparation is in your lab, just take a look, try to remember these two systems. It's very easy to prepare the system in your lab because regular silica gel with a mixture of water and acetonitrile, or regular cellulose with a mixture of water and acetonitrile will provide you this very nice separation. You see, very sharp peak with a very, very good resolution between different forms in your preparation, and it's very fast. You don't need to make some complex buffers, wait a lot of time for measuring. You just make this analysis as fast, and then it's very useful in your own lab. And surprisingly, I told you that it's very difficult to measure the radiolytic degradation of your product with TLC. In the case of PSMA ligands and in the case of this system, it's possible to identify some side products of the radiolysis even with TLC. And these results will be very good correlate in good agreement with HPLC results. So just to be sure that your preparation with lutetium is good enough, you may perform the system. You will see the labeled compound, the desired chemical form, or if it takes some radiolysis action, you will find some side products, and then you could determine the radiochemical purity of your preparation correctly. Yeah, this is by the way, the correlation between HPLC and TLC in the case of lutetium in the terms of this side forms produced during the radiolysis. This is not unbound lutetium. Now I'm talking about the forms after the radiolysis of the molecule. So identify or not identify? This is the question. From the one side, yeah, of course, we are talking about medicine of 21st century and so on. So the quality of our preparation should be as high as possible. Another problem is analytical problem. We see the separation parameters like this. We see a lot of side forms. Just at the beginning, when you develop your method, you could say, oh, okay, this forms is unidentified. We just make it like a sum of impurities and exclude it from the radiochemical purity value. But in fact, I told you that we, under the pressure in the legislation on the Ministry of Health, of the pharmacy requirements and so on, in fact, we have to identify these impurities and we have to establish limits for these impurities. Yeah, it's very easy to say, oh, the radiochemical purity should be about 95%. But where the proof for this value? Why it should be 95%, not 98%, or maybe not 91%? What is the reason of this value? So that because all these impurities should be identified and with established limits. But in fact, of radiopharmaceuticals, it's very, very hard work. Sometimes it's even impossible. There is no standard samples for the radiopharmaceuticals, of course. For starting materials, yeah, it should be. You could find it. For the already prepared radiopharmaceuticals, no, nobody gave you, okay, man, this is the sample of your PSMA. It's perfect. The radiochemical purity is 100%. Just use it for comparing. No, there is no standard samples. And any analytical chemist will ask you, and what should I do with these peaks? How I should make I'm sorry, hard morning. What should I do to qualify it and quantify it? So the problem is purely analytical and under the legislation of the pharmacy field. So, as I told you already, this is on your own responsibility. Maybe the good decision is to use, for example, two methods in your own lab. One method, it's very good method, which can provide you all the side products. And during the research and development of your product, you use this method. Then you optimize your synthesis process, obtain one good peak, and then to the quality control datasheet, you put another one method, which is much more easy. It's much more easy to perform without any side products. Maybe this is one way to find a good decision in this problem. I didn't say another way, just to be honest, but just to remember about that depending to methods, a lot of impurities may be identified or not may be identified, and it's your own responsibility during the quality control. And at the end of this lecture, let's talk about very popular part of radiopharmaceuticals is alpha-targeted radiotherapy. Why it's popular? Oh, come on, radiochemists.
That's patient distance, and it can target the tumor specifically if you administer it with the right organ.
Yeah, it's very low penetration. Yeah.
For the other tissues.
In theory, yes, it's very low linear energy transfer, providing double point destruction of DNA, which is more efficient for the cell deaths. Yeah, for the cell death. But all these parameters is very good in theory. We are talking about alpha emitting radionuclides. You are radiochemist, yes? No, I'm sorry. Okay. What is the biggest problem of alpha emitting radionuclides? I believe for any kind of, but except for the alpha emitting radionuclides. No. Just think about the qualification, the equilibrium, and something like that. The biggest problem is daughter radionuclides, because there is no alpha emitting radionuclides suitable for the therapy or suitable for the synthesis, which form the stable form with the one alpha decay. We have on one alpha decay, we have the stable daughter, and no, we have the family of daughter radionuclides, which is in equilibrium, which is absolutely different chemical elements. Yeah, and they have different biodistribution, different chemical properties, so different type of radiation. It may be alpha decay, beta decay, gamma decay, again alpha decay, and so on, so on, so on, so on. This is the biggest problem of the alpha radionuclides, and for me, it's the biggest limitation today for the implementation, the big implementation of the alpha radionuclides into the clinical practice. Yes, of course, I believe everyone saw this nice pictures for the radionuclide therapy of the patient. Here, it was a man patient with a prostate cancer. After 2 circles of lutetium PSMA, there was no response to the therapy, but only after 2 courses of alpha therapy with PSMA, yeah, we see almost complete response to the therapy. It's very nice. In theory, it's very nice because today physicians found this effects of the insensibility of the tumors to the beta therapy. Do you know about it? If we perform the radionuclide therapy with beta emitters, then very often we could see the not stable remission, but the disease comes again to the patient. And in that case, beta emitters is not efficient. Only alpha emitters may help to the patient. So this result is very nice from the one side. Let's return to the chemistry of the alpha emitting radionuclides, and particular to the actinium decay. So you're not radiochemist, but maybe you know about the actinium decay. Somebody? Francium? Yeah, the first daughter is francium. The second daughter is astatine. Bismuth, yeah. Bismuth 213 is also used in the targeted alpha therapy, but the half-life is very short. It's about 46 minutes or something like that. And this half-life is not enough to correspond the desired dose of the radioactivity to the tumor. But anyway, if we prepare the radiopharmaceuticals with actinium, we always have all these daughters in our reaction mixture. Yes, this is the one problem. Another problem, how to, if we talk about the radiochemical purity, yeah, we have the same reasons just to determine it. We have to determine the actinium, which is unbound. We have to determine actinium, which is conjugated with the vector molecule, and also maybe it's good, but it's not always possible. Determine some side products of your synthesis of the radiopharmaceuticals. But what the real problem is, actinium itself, it's invisible. Except alpha particles, it's very difficult to determine it radiometrically. How we can measure the radioactivity of the activity then? What do you think about it? How we could measure the distribution of actinium activity? In the case of TLC, for example. Oh, come on.
Peaks of particles have discrete spectrum.
Alpha spectrometry in the case of the such kind of radionuclides, especially in terms of quality control of radiopharmaceuticals, it's not applicable. It's very difficult to perform. You may perform alpha spectrometry for some samples of the air, water, earth, or something like that. But when you have to quantify the real fraction of your radionuclide with a TLC, for example, method, no, it's not suitable. So the only way to measure the activity of actinium is just to weight the equilibrium of actinium with his daughter radionuclides. For example, francium. The francium has a gamma line, so it could be easily quantified. And then with bismuth, also had the gamma line, it also could be very easily quantified. Here, these daughters is very short-lived. They will decay absolutely immediately. So we're just talking about here with the main daughters is francium and bismuth. Then, keep looking. The francium has a half-life about 5 minutes. The bismuth has a half-life about 45 minutes. And all this radionuclide is present in the starting solution of your radiopharmaceutical preparation. Francium is from the group of natrium. It's absolutely different kind of radionuclide. So its chemical behavior, it's different to the actinium and even bismuth. Here, the equilibrium time between actinium and francium is about 1 hour. 1 hour, maybe less, maybe 55 minutes. Then we will reach the secular equilibrium between actinium and francium. The secular equilibrium between actinium and bismuth takes around 6 hours. The full equilibrium of actinium with all his daughter takes about 20 hours. And then you just remember that we have problems with the radiochemical purity determination. We have problems with radiolysis because this is alpha emitter. The radiolysis grade is even higher than in the case of the beta emitting molecules. Yes, we have to determine the radiochemical purity of that kind of preparation. We make the synthesis, make the mixture, heat it, put the spot on the TLC plate, make a separation. And what we have to do now? We have to wait at least 1 hour just to reach the equilibrium with francium, just to detect it. But in the starting solution, we also have the bismuth and also have the francium from the beginning of the synthesis. And all these parts are now just distribute on the plate. So we have to wait the decay of the free bismuth, free francium, accumulation of the equilibrium francium and equilibrium bismuth, and only after this we may perform the measuring of the TLC plates. So how long do we have to wait now? At least 1 hour if there were not big amount of bismuth in your starting material, if it was purified somehow by cation exchange or solid phase extraction or something like that. Or you have to wait sometimes even 24 hours just to perform good measuring, just to perform the good distribution of your plate. I have somewhere here this is example of the distribution on TLC plate of the actinium preparation. This is a distribution right after the development of the TLC plate. This is the same plate measured 2 hours after the development of the TLC plate. And this is after 22 hours after the separation. You see the picture changed dramatically. So at the beginning of your preparation, at the big starting time point, it's impossible to quantify the fraction of your radionuclide with your desired chemical form. Only on the next day, we may measure our TLC strip and then quantify and calculate the radiochemical purity value. But what does it mean? In the practical reasons, we make the preparation, we put the spot, make the TLC plate, we wait 24 hours, then we measure, we calculate the radiochemical purity. But this value of radiochemical purity is for the just day after your day before your time point. So for this moment of time, it's not the same. It may degrade radiolysis, any other processes, release of the radionuclide, and so on. So, in my opinion, this is the biggest limitation. So we cannot perform the radiochemical purity test for alpha emitting radionuclide, especially like actinium-225, just at the time of the planet injection to the patient. Yeah, at the time of injection, we don't know the real radiochemical purity of your radiopharmaceutical. Of course, you may perform a lot of test synthesis, make the development, validate your development and synthesis procedure just to say, oh, our synthesis methods just guaranteed that our radiopharmaceutical will have the radiochemical purity enough for the patient. But it's only words. Where is the measuring results? Where is your real chromatogram? Where is the real radiochemical purity value? So that because even with this nice results of the therapy of the patient, even with all this promising results published in different articles, we have to be very careful before the real implementation of the alpha-emitting radionuclides into the routine clinical practice, especially like actinium conjugated with some vector molecules, and bismuth conjugated with some vector molecules, thorium-227 conjugated with some antibodies and vector molecules. Today we have only Xofigo, the first registered alpha-emitting radiopharmaceutical on the market. But what is Xofigo? It's radium chloride. It's a solution, just a solution of radium chloride. Radium accumulates in the bones, in the metastasis in the bones, and we see the alpha treatment of this metastasis in the bones. Yeah, it's very simple. But in the case of much more complicated radiopharmaceuticals, in the case of the vector radiopharmaceuticals, we have the problems like this. And another problem is, okay, even if we could measure the TLC plates on the next day after the separation, what about the HPLC? I told you about all the side products, thermocyclization products, but here, because actinium, it's not visible for the detectors, we cannot perform. We cannot perform the HPLC. If you inject your preparation with actinium into the HPLC, you will see activity in the dead volume, nothing more. Yeah, because it's impossible to reach the equilibrium between radionuclides in dynamic mode. Yeah, we have to wait at least 55 minutes or even 6 hours. Of course, if you have I don't know, if you have in your lab some bad PhD student, you could say him sit close to the chromatograph, collect all the fraction drop by drop, and then measure it with some appropriate detector on the next day. And in the Excel or in the Origin, just to make this kind of HPLC chromatograph, but it's impossible. I don't know. It's very difficult. This drops collection, this measuring on the second day after the analysis. Yeah, it may be interesting for your PhD thesis, but for the practice, it's about nothing. So I don't have answer how to solve this problem so far, unfortunately. I just want to put your attention on this moment. If you will one day works with alpha-emitting radionuclides into your practice with radiopharmaceuticals. But of course, quality assurance, the good synthesis methods, good analysis method, carefully found and carefully developed analysis methods will provide you best results with the synthesis, with quality control, and of course with the human use of obtained radiopharmaceuticals. If you have any questions, I will try to answer. Thank you so much.
But I didn't understand why the TLC of the 24-hour solution is going to show the radiochemical purity of the compound the day before, because when you develop it, it's in its current state where it has undergone radiolysis and other stuff. So the current state will be revealed.
Our starting point is 0. Let's call it end of synthesis time. Here we make our solution which contains actinium. Yes, this is the starting point of your solution. You take a part of the aliquot of your solution, put it on the TLC plate. divide, and then you start to wait. It sounds perfectly, it's wonderful. So we wait 2 hours, 2 hours, 2 hours, 2 hours, 2 hours, 1 day. Then next day, you measure this, and you obtain the result of this already separated state. So this picture corresponds to this time point, not this time point, and you obtain the result. So for example, 90% of radiochemical purity corresponds to this time point. But you already here in the second tape, so you don't know the real picture, what's happened during this day of reading in the configuration, release of the activity. So if you want to know about the current situation at this time point, you also put another drop, develop another TLC plate, then you will play another plate. Here you will identify the situation that day before your day, but what about the current situation?
I'm not. I didn't understand the second part as well. Why wait another day if it's already in equilibrium? Why not develop it as it is?
It's the region complex integrity. But what's happening with your depth model? You perform the synthesis of actinium with some ES and A. Okay, at this time point, everything is okay. All actinium is communicated with the depth. But during this time, while the agent, some part of actinium, some part of PSMA may be destroyed by radioactive action. So, in this, then after you perform the TLC, the situation changed. All these parts in the equilibrium, but you know, it's just kinetically, it's not, I don't know, it's not destroyed at one moment, and then but this they will already have the equilibrium with all these possible species. You know, because the destruction is also time dependent. So it's change. It's not something like that. Yeah, if it's something like that, for example, on this line. Okay, we will wait a few hours and then perform the separation again, and then you could say, okay, this type of map is this type. No, change. And also, always remember that we perform the distribution on the plate. Yes. I will show again the current. All the radiant lights in equilibrium in the middle, of course. But all of this radiant light have different distribution on the plate. So even if they under equilibrium, some parts of equilibrated bismuth will stay in the ore. Some part of the bismuth will incorporate in the molecule. So we often develop again the equilibrium of the plate, not in the solution. Clear?
Yeah, it's a lot more clear now. Thank you.
We could discuss other things, right?
Ну, тогда без микрофона можем ставить. Окей, тогда без микрофона. Да, так.
Иван, мы через 2 минуты готовы начинать.
Так, здравствуйте. Вы меня слышите хорошо?
Да, слышим хорошо, видим презентацию.
Ну, тогда я, в принципе, готов. Ждем, получается, пару минут.
Да, да.
И начинаем.
Мгм. So we are ready to start our lectures on the second half of the day. And the first lecture will be given by Ivan Ushakov from Tomsk Polytechnic University. So Ivan, please, the floor is yours.
Thank you very much. Good morning, dear participants. My name is Ivan Ushakov. I am representing Tomsk Polytechnic University. Can you please have in mind that I am not a native speaker of English, therefore I can have some mistakes and mispronunciations. Now I'm going to tell you about the production isotopes for medical purpose. A nuclear reactor is an apparatus in which a fissile material can be made to undergo a controlled, self-sustaining nuclear reaction. The reactor produces fast neutrons which are slowed down in the moderator, and the slowed down neutrons start new fissions. As everyone knows, the basic equation for the fission of uranium-235 nuclei under the action of the neutrons is shown on the slide. This is the main nuclear reaction after which several fragments are formed and energy is released. By regulation this nuclear chain reaction, there will be a steady state production of a thermal neutron. Let us consider the classification of nuclear research facilities. They are subdivided into subcritical stance, critical stance, and research reactor. A research reactor is a reactor designed for fundamental and applied research. All research reactor can be divided into two types. The first of this is a beam nuclear reactor. In a beam reactor, the maximum flux density is concentrated in the beam. The second one is a material science reactor, where the maximum neutron density is concentrated in a certain volume. Any research nuclear installation can be divided into three groups. These first groups with a capacity of up to 100 megawatt. For example, it used for testing materials and equipment for nuclear power. The second group is those reactor which has the power up to 20 megawatts for education purposes. It used for education purposes, for example, fundamental physical research and production of radioactive isotopes. And the third group with the power up to 1 megawatts. Usually, only research reactors are flexible enough for use in radioisotope production. Currently, there are about 223 operation research reactors in the world. Approximately 83 reactors are used to produce isotopes for medicine. The main important characteristics of a reactor is a nuclear neutron flux. The higher the neutron flux, the better. Also important characteristics are the neutron spectrum and temperature at the irradiation position. The distribution of reactors by neutron flux is represented in the table. Keep in mind that only 4 reactors have a continuous neutron flux more than 10 power 15. On the slide, you can see how research reactors are distributed around the world. For example, in the Russia and the USA, there are greater number of the reactor than in other countries. China is rapidly building the reactors too. Here, the name of those reactors which has a most high flux reactors in the world. In the tables, you can see their locations and parameters like power, maximum flux density for thermal neutrons and for fast neutrons. You can see also the main isotope produced on this high flux reactor. For example, for medicine, in SM3, you can see that produced lutetium 177. Also, you can see the tungsten 188, iodine 131, for example, and others. Let's consider some theoretical aspects. Neutrons are divided into 3 energy groups. This is a thermal, epithermal, and fast neutrons. Thermal are those that are in thermal equilibrium with the molecules, atoms of the environment. Epithermal neutrons are intermediate energy neutrons, usually in the kilo electron volts region. Fast neutrons are with high energy. They have an energy distribution similar to that of fission neutrons. As I mentioned before, the neutron spectrum is an important characteristics. On the right, the example of neutron distribution is given. On the left, for conventions, the continuous spectrum is divided into intervals named group. Each group contributes to isotopes production, for calculations. There are two main ways to determine the distribution of neutron flux spectrum. The first one is a theoretical, the second one is practical. For theoretical determination, one can use such program as MCMP or MCU or others. You can see the view of Monte Carlo Universal Program, it's MCU program, and the listing of this program. Here you see the distribution of neutron in different channels in Tomsk reactor ERTT, which we calculated using this program. On the right, distribution of the flux in the channel covered with a boron carbide is represented, and in the left part, in the central channel of our reactor. For practical determinations, the activation kits are used. Usually, there are various elements with a known number of nuclei. Each item of the kit can determine a part of the spectrum. Also, the part of the spectrum can be found using the capture spectrum material, for instance, cadmium, for cadmium difference. After that, you activate your kits. You can use the gamma spectrometers that found the activity of significant nucleus. Also, you can use the other programs, for example, Feed, Serpent, MCMP, and MCU, that to found the activities of your samples after the irradiation. The examples of feeds programs you can see on the slide. Let us consider the most common reaction types for the production of medical radionuclides. The first type of reaction and gamma reaction is used in most reactors. In some cases, the reaction and gamma produce a short-life radioisotopes that decay by beta radiation into another radioisotopes. The product can be chemically separated from the target. This will make it possible to obtain radioisotopes with a high specific activity or carefree radioisotopes. For example, if you in the slide shows some examples of reaction from the previous slide. According to the first type of the reaction, it's possible to obtain radionuclides with a carrier. This type can be used to obtain radionuclides such as a gold, molybdenum, and lutetium and other. For example, when you irradiated target made from the lutetium, you obtain the lutetium-2 with a care added with a K rate. According to the second type of reaction, radionuclides are produced without a carrier. This type can be used to obtain radionuclides the same like lutetium 177, but in this case you irradiated the ytterbium target, which converted into ytterbium 177, which decays, disintegrated into the lutetium 177. In a NP reaction type, the absorption of a neutron leads to the emission of a proton in the form of the escaping particle. This reaction is caused by fast neutrons having an energy above a certain value known as a threshold energy. The core of the product can often be chemically separated from the target. This reaction can be used to obtain radionuclides such as scandium-47, phosphorus-32, and others. An alpha reaction is also threshold reaction, since neutrons having an energy above a certain value, threshold energy, are absorbed by nucleus, causing the emission of an alpha particle. In some cases, the reaction produced proceed with the thermal neutrons. And certain radioisotopes are produced by multi-step processes or sequential neutron capture. An example is obtained tungsten 188 from the tungsten 186. And the last reaction to consider is fission reactions. The fission of uranium-235 induced by thermal neutrons produced many useful radioisotopes. Each division gives 2 division fragments, a light group and a heavy group. The example is the production of strontium 90, molybdenum 99, and others. On the slide you can see the equations which you can use that to found in first approximation the activity of your target. The equation has 3 significant parts. First of all, it's accumulation part, disintegration part, and burn up of your isotopes. If you crossing the burn up, you can to obtain the differential equations, and after that you can look at this production between cross-section of the target length. You can to use a more simple, you can to simplify your equation to the thin target or thick target. In the practice, the activity produced in the target by irradiation will be less than the activity calculated using the equation above, due to several factors such as self-shielding effect, reactor power, change in the neutron flux due to neighboring samples in the reactor, target field burn up over time, and destruction of the product core due to subsequent neutron capture. Self-shielding effect is significant in the case of the target with a large activation cross-section when the neutron flux in the target volume decreases, which reduced the increase in activity. You can take in the account self-shielding effect for thermal neutrons if you use this equation that to found parameters of the self-shielding. For resonance neutrons, it's a little bit different. You need to use a resonance width reaction and total resonance width that to found the self-shielding effect for resonance neutrons. And if you draw graphs, you can see that when you increase dimensional parameters, your neutron self-shielding factors for resonance and for thermal neutrons are different. Don't forget about accumulation of isotopes at different neutron fluxes, because if you have big fluxes, you need don't forget about a burn up, because after the increasing of the activity, you obtain the decreasing of the activity because you have a burn up of the target and disintegration, of course. And the choice of the packaging materials and sealing methods will depend on various factors such as the physical form of the target, target characteristics, duration of exposure, the design of irradiation position in the reactor, and use after the irradiation. For radionuclides, for medicine radionuclides in the reactor, and commonly use packaging in aluminium, and also use a packaging in titanium capsules and graphite capsules. On the slide, you can see the laboratory facilities for radioisotope production. It's a typical facility could consist of all of the following specialized facilities and capabilities. You can see pictures of disposable aluminum container and reusable urns. Disposable containers are usually sealed and can accommodate a quartz ampulla with a target like a photos. The quartz ampulla can be open or sealed. After the irradiation, the container can be delivered to the hot cells in the lead transport protection. The slide shows photographs of the container for irradiation of needles or iridium disc. Typically, the targets after being irradiated are moved to hot cells for processing. They can be directly loaded into the hot cells using special channels into the hot cells, or can be transferred using a pneumatic mail, for example, or a shipping container, for example, like this on the photos. Samples are usually pulled from the core using a reel with aluminum wire on example is shown on the photos. Since samples are usually highly radioactive, distance rod or grips are used to move them to the special hot cell channel or to the transportation containers. To work with the high radioactive samples, hot cell made of lead or heavy concrete are usually used. Operation with the target are carried out using manipulators. The target is usually observed through lead glass or a video camera. The chamber must contain a collection of the solid and liquid waste, as well as any technological devices. On the left photographs, you can see of the hot cell made of the lead, like this. On the right photos, you can see the hot cells which made from heavy concrete. The main types of manipulators for working in a hot chamber are as follows. These are robotic, master slave, or sword manipulators. The sword manipulators is the simplest and cheapest. It is a rod threaded into a ball socket and the end of which there is a grid in the end. But it is not easy to use compared with to a master-slave manipulators, which has more degrees of freedom. The aluminum container can be torn, untwist, cut, dissolve. I mean, when you put it in the hot cells. After this, the target, for example, in quartz ampulla can be opened. On the slide, you can see photographs of devices for opening containers. For example, this is cutting machine. This is machine for break of the aluminum container. On the two part, this is top part and the bottom part. And after that, you simplified get the quartz ampulla for next operation. The ampulla inside the hot chamber can be opened in several ways. First of all, is a breakdown of the bottom of the ampulla, crush the ampulla, and the third it's open with a cutting machine. After the ampulla is destroyed, this is destroyed machine, the target is dissolved along with the glass. Therefore, the ampulla must be clean on the outside to reduce product contamination. When cutting or punching of the bottom, a small amount of glass reaches the target. Let's look at the production of radionuclides at the research reactor of Tomsk Polytechnic University. This reactor in Tomsk has 6 megawatt and the maximum neutron flux is 2.1 by 10 power 14. Other characteristics are shown in the slide. The exterior of the reactor building are shown on the photos on the left. The control panel of a nuclear reactor is shown on the right. The appearance of the reactor is shown on the photo on the left. When the reactor is operating, a beautiful glow appears called the Vavilov-Cherenkov glow. And the slide shows photographs of the reactor fuel assembly for our reactor. The enriched uranium is contained within an aluminium shell. The side section, the active zone, the upper platform, which the samples are loaded to the nuclear core. Nuclear core in these pictures is red color. And you can see the layout of 6-pipe fuel assembly named RTT3M. And also you can see the cartogram of our reactor core. The red color boxes and the yellow color is a fuel assembly. The gray color box color is a beryllium box. And in the central of our nuclear core we have a central capture, and in these 4 experimental channels we have the maximum neutron fluxes. Also we have one of the vertical experimental channel inside of the fuel assemblies that we can to get the fast neutrons. And the others experimental channel are used for several experimental work for neutron activation analysis and for other papers. A photograph of the core is represented on the left. You can see the many tubes going to the active zone from the top to the bottom, as well as from the sides. Most of them are experimental channels in which isotopes are irradiated. The cartograms also you can see on the right, and also you can see the view of the core, and this is assembles. This is a beryllium block, and this is fuel assembled. This is photos made from the loading of the nuclear core. Okay, the most popular isotope, molybdenum-99, is usually obtained from the fission reaction of uranium-235. We get molybdenum-99 from the reaction of radioactive neutron capture. We irradiated molybdenum oxide in sealed container. When irradiation natural molybdenum in a thermal neutron flux 10 to the 14 power for 100 hours, the molybdenum-99 yield does not exceed 3 curie per gram. When enriched molybdenum is irradiated in the same neutron flux, the yield of the molybdenum-99 is about 15 curie per gram. You can compare it with those value which made from the fissionable reaction. And you can see the irradiation devices when you irradiated uranium dioxide. The main disadvantages of the activation molybdenum sorption generators are low specific activity and a big size of generators. While when producing fusion generators, the main problem is a large amount of radioactive waste. The slide shows a device for isolating the most popular medical isotopes, technetium-99m. The device is the technetium-99m sorption generator. Its heart is an aluminum oxide. And doctors wash technetium-99m out of the generator every day with a sodium chloride solution. The finished eluate enter a sterile evacuated bottle at the vacuum. I mean, here you see the 99m technetium generator kit with a shipping container and a sorption column with a lead protection. Such generators are produced at Tomsk Polytechnic University and supplied to medical institutions. The elution curve show that different volumes of eluent are required to completely flush out all accumulated technetium-99m in the generator. So for a standard generator produced by irradiating uranium-235, 3 milliliters of eluate are required, and for a generator made from activated molybdenum-99, 7 milliliters are needed. This is due to fact that molybdenum-99 activation requires a larger column to load a larger mass of molybdenum-99. And you can see the different generator columns and the operator rooms, an external view of the hot cells made from the heavy concrete. TPU use a very simple scheme for the production of technetium-99m generators. The scheme includes the following operation: irradiation of the target with a molybdenum oxide in the channel of a nuclear reactor. After that, transporting the target to the production site, opening the aluminum case and dissolving using an opening machine. You can see on the photos. After that, we dissolve in molybdenum oxide using alkaline solution, neutralization with hydrochloric acid, and finally sterilization and loading of molybdenum-99 into the generator column. Loading is carried out using a dispenser system. You can see on the schematic. This simple technology makes it possible to quickly and efficiently produce a large number of generators in a short time. And also you can see the generator before assemble and generators on the loading position. This slide provides data on the advantages of an activation generators over of uranium-1. The yield of the technetium-99m more than 95%. The generator is kept dry between elutions to reduce radiolysis. The loss of expensive molybdenum-98 is less than 8 percent. This is achieved through regeneration from the spent generators. Radioactive waste includes only the sorbent, ampoules, and containers for molybdenum irradiation. In contrast to uranium technology, where a large amount of radioactive waste, which requires burial, is generated. For example, iodine, cobalt, and etc. Fission fragments. This technology makes it possible to produce generators with activity from 5 to 30 gigabecquerels. Another advantage is the absence of long-life radionuclides in the generator. Another way to obtain technetium-99m is extractions. TPU has technologies of an extraction technetium-99m generator. The extraction technetium-99m generator can be both stationary. It used for simultaneously provision of technetium with drugs in large cities, for example, and transport to individual clinic also. Generators have full remote control. The generator has been working without failures at the ERT reactor for about 30 years in the made of daily deliveries of drugs to the clinics of the Tomsk. The external of these generators you can see on the photos. The scheme of the technetium-99m extraction generators with a multiple extraction cycle includes the following stages. First of all, it's a formation of the initial aqueous phase, I mean target dissolution, introduction of salting out agent. After that, methyl-ethyl ketone extraction of the technetium-99m using this extraction column. After that, extraction separation, evaporation to dryness, and dissolution of technetium-99m in physiological solution. After that, of course, packaging in bottles and sterilization of the drugs. On the photos, you can see the ampoules before irradiation of the molybdenum with a natural enrich. The main advantages of the technology are extraction technology is concentrating high bulk activity of the technetium-99m, use of cheap molybdenum oxide with a natural isotopic composition, low cost of radiopharmaceuticals, and high chemical, radiochemical, and radionuclidic purity. The third installation in this series of generators is the technetium-99m extraction chromatographic generator. The technology includes both processes used previously, sorption and extraction. This is extraction, this is sorption. It can be transported over long distances as a sorption generator. It can also work on natural cheap raw material as an extraction generators. The generators eluate containing the radionuclide technetium-99m is used in medicine for diagnostic purposes, mainly for functional diagnostic of the thyroid glands and salivary glands to diagnose other organs. Technetium-99m and other radioactive isotopes must be mixed with targeted chemicals. Here we present radiopharmaceuticals for diagnostics and therapy that we have developed and put into production with the technetium and iodine. Now, let's say a few words about the development and production of microspheres and radiopharmaceuticals for the treatment of cancer. Microspheres with yttrium-90 radionuclide are used to treat liver cancer, but the way this is the only cure for this disease. This drug has a double damaging effect. Firstly, the drug clogs the artery that feeds the tumor. Secondly, beta radiation additionally affects the tumor. All this leads to its destruction. During production, yttrium, aluminum, silicate microspheres are firstly irradiated in the reactor channel. You can see the photos when we use lab balances that weight it, and then they are packaged into certain doses. Each dose is selected for each patient individually. We also wash microspheres after irradiation that to decrease the contamination of the cover of the microspheres. The finished dose of the yttrium-90 is administered to the patient through the femoral artery. Radioembolization is carried out using a plexiglass box for radiation protection. The pictures on the right shows the distribution of the microspheres in the vessels around the tumor. The slide shows the characteristics of the finished drugs. The wide range of the activity indicates that different drugs may be used depending on the size of the tumor. Okay, next, radioisotopes is lutetium 177. Lutetium can be obtained by direct methods. To do this, enriched lutetium is irradiated with a neutron flux. As a result, lutetium with a carrier added is obtained. The main disadvantage of these methods is the presence of the long life lutetium 177M and low specific activity. Lutetium with high specific activity can be obtained by irradiation of enriched ytterbium. This indirect methods of obtaining is called no-carrier added. The main disadvantages is the difficult procedure for separation lutetium and ytterbium, as well as the small reaction cross-section. The next point to consider is a chromatographic methods for obtaining lutetium without a carrier added. A radioactive mixture of lutetium and ytterbium adsorbed at the top of the column. The lutetium and ytterbium are then eluated with isobutyric acid or other organic acids and fractionated. The cementation methods and the extraction chromatographic methods are also used to separate lutetium and ytterbium. The cementation methods use sodium amalgam with the help of which only ytterbium is reduced. Extraction chromatography can only elate lutetium, but it is quite expensive. The slide shows photographs of the electrochemical cells for cementation and the column for chromatography, extraction chromatography. Next isotopes is iodine 125. Also, the most popular radionuclides for the treatment of tumors is iodine 125. Microsources are supplied sterile in a protective cassette, which ensures almost complete absorption of low energy and gamma radiation. The number of sources used in one operation usually doesn't exceed 75 pieces. The nuclear physical characteristics of iodine 125 are optimal for its use in the treatment of prostate cancer. The production of the iodine 125 isotopes itself includes the following steps: placing xenon gas in the stainless steel ampulla, irradiation of the ampulla in the reactor, rapid placement of the irradiated ampulla into the hot cell, opening the ampulla, freezing irradiated xenon, after that xenon exposure, and separation of the target product into a new ampulla, and use of the resulting iodine 125 as a raw material for the production of the microsources. Ready-made microsources are cylinders made of medical titanium. Inside each cylinder, there is a gold wire, an X-ray market, and iodine radionuclides in the form of silver iodide located in a porous aluminum oxide ceramics. And also you can see the microsources on the photos of the microsources on the hand, and the line for production of these microsources from the iodine and capsules. This slide shows images of the manufacturing process of finished iodine 125 cartridges at Tomsk Polytechnic University. Microsources with iodine alternating with a plexiglass spacers are placed in a thread. The thread is then placed in the cartridge. Before the operation, doctors insert a thread into a needle for implantation. It should be noted that the production of sources is high tech. The doctors implants up to 75 sources. The activities of these sources are selected depending on the prevalence of tumor. Okay, let's have a look at the next point of our discussion. Phosphorus without a carrier can be obtained by irradiation of sulfur with fast neutrons. On the slide, you can see the hot cells in which the radiochemical separation of phosphorus and sulfur is carried out. Sulfur is sublimated in the vacuum, and after this phosphorus is dissolved and purified on ion exchange resin. You can see on the right part of the slide the column with a cation and anion exchange resin. And also when you switch off the light, you can see the Cherngov glow on the column. To obtain iodine, a tellurium target is irradiated. Iodine and tellurium are separated by dry distillation. To do this, the target is poured into a tube and heated in the vacuum. The evaporated iodine is sorbed on the charcoal column. After the cooling, the iodine is eluted from the column using a sodium hydroxide. Using these methods, you can obtain iodine 131 from the tellurium target. And you can see the ampulla with tellurium oxide, cross-section of the ampulla with the tellurium oxide. Operation of the production site requires the following licenses and permits. For example, licenses of Rostechnadzor, licenses of the Ministry of Industry and Trade for the production of drugs, Rospotnadzor, certificate of compliance with the requirements, certificate of the GMP, and others documents. Thank you for your attention. I hope that you have a good experience with my presentation. If you have any questions, I would be glad to answer them. Of course, you can type it and send to my emails, and I answer you later if you want, of course. Thank you for
Thank
your
you very
attention.
much. Thank you. So questions? No? Oh, one question from me. When you open these vessels, how dangerous is this operation?
You mean when I open the quartz ampulla?
Quartz and metallic, yeah.
Yes, it's very dangerous because when you open this ampulla, you can pour all your target on the hot cells, and after that you need to irradiate again. Therefore, a lot of labs using a crush the ampoulas because it's more safety. You cannot to loss of the target.
Okay, thank you. Any more questions? Okay, if I don't see also online questions, only the words thank you for your great presentation. So once more, again, thank you and goodbye for today. Yeah.
Goodbye. If you have any questions, please send to my emails all your questions.
Yeah.
Goodbye.
Bye-bye.
Goodbye.
Bye.